Good evening, Dr. Lucas. Hello.
Hello. In Poland, it would be good afternoon now, but still, hello.
Oh, nice to meet you.
Nice to meet you too. I'm very pleased to be here with you today.
What time is it in the Poland?
In Poland, we've got 2 p.m. currently.
Oh, yeah, yes. Thank
Okay.
you for the speaker at guest lecture our campus.
No, it is a pleasure for me. I would like to thank you. It's very
Yes,
nice. It's for me, it's the first time I'm giving a speech in Indonesia, so it's like an honor for me.
yes. Okay,
Okay, thank you, sir. We may begin.
okay, yeah.
Yeah.
Maybe MC, please.
It's me, sir.
Oh,
I'm Professor
moderator.
and a
Yes,
moderator. Okay.
yeah, thank you.
Yeah. Yes. Good afternoon, Professor Lucas Sviantek. Did I right to pronounce your name, Sviantek, right?
It's perfect. It's
Oh,
perfect.
perfect. Okay. Thank you. I just learned it from I learned it from the internet how to pronounce you. Your name seems familiar for just like the famous tennis player Ian Swiatek, right?
Very similar, yes.
Very
Świątek,
similar,
exactly.
yeah. Okay. Yes, good afternoon for Professor Swiatek, and good evening for all lecturers, students, and all participants in in the Zoom meeting. Assalamualaikum warahmatullahi wabarakatuh. Yeah.
Waalaikumsalam.
Peace and blessing be upon you all. Okay. First of all, our distinguished guests, the Honorable Dean of the Faculty of Pharmacy, Universitas Pancasila, Professor Samsudin. Our esteemed speakers today, Associate Professor Dr. Lucas Fiante from the Department of Virology, Diagnostic Laboratory, Faculty of Medicine at Medical University of Lublin, Poland, and our respected lecturers, academic staff, students of the Faculty of University Pancasila. We warmly welcome all of you to this our special international guest lecture. Let me introduce myself. My name is Gumilar Adinugroho. I'm a lecturer at the Faculty of Pharmacy Universitas Pancasila, and I will serve you as the moderator for today's event. Right. Our theme for today's lectures will be the antiviral potential of natural products. But before of that, I would like to please allow me to share some a few technical reminders to ensure a smooth session. First of all, please keep your microphone muted during the lecture to avoid the interruption. Second, you're welcome to turn on your camera on, especially during the question and answer session. And if all participants have any questions during the talk, feel free to type them in the Zoom chat box. We will address them during the Q&A session at the end. Okay, for the next agenda, we would like to listen the welcoming speech from the Dean of Faculty of Pharmacy, Universitas Pancasila, to Professor Samsudin. We respectfully invite you to deliver your speech. Please, sir.
Thank you, Mr. Gumilar Adi Nugroho. Good evening, everyone, and good afternoon, Dr. Lucas Wetsik. Assalamualaikum warahmatullahi wabarakatuh. The Honorable Associate Professor Dr. Lucas Wetsik from Faculty of Medicine, Medical University of Lublin, Poland, as speaker. The Honorable lecturer, student, staff, and participant. As the Dean of Faculty of Pharmacy Universitas Pancasila, I highly appreciate the presence of Associate Professor Dr. Lucas Wetsik from Faculty of Medicine, Medical University Lublin, Poland. Since the ancient times, natural products have served as a major source of drugs. About 50% of today's pharmaceutical drugs are derived from natural origin. Viruses have been resistant to therapy or prophylaxis longer than any other form of life. Currently, there are only few drugs available for the cure of viral diseases, including acyclovir, which is modeled on natural product parents. In order to combat viruses which have devastating effect on human, animals, insects, cropland, fungus, and bacteria, many research efforts have been devoted for the discovery of new antiviral natural products. I go to know Associate Professor Dr. Lucas Wesit, one of the experts in the field. I hope the truth, the discussion, the emergence in this guest lecture, this can encourage our students to move further in studying development in the upstream pharmaceutical sector, namely the development of medicinal plants that have the potential to be antivirals. Finally, I would like to confer my congratulations again to the audience for holding this guest lecture. Hopefully many ideas can inspire the audience through today's guest lecture and become a blessing of knowledge for us to put into practice together. Wassalamualaikum warahmatullahi wabarakatuh. Thank you.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you very much for Professor Samsudin for your thoughtful speech. Okay, before we begin the main lecture session, allow me to briefly introduce our honorable speaker, Professor Lucas Viantek. Can the committee show the CV? Okay, yes, it's my great honor to introduce our distinguished speaker, Associate Professor Dr. Lucas Viantek from the Medical University of Lublin, from the Department of Virology and Viral Diagnostic Laboratory. Professor Viantek's teaching activities including the microbiology, oral microbiology, and then the infectious disease, and also the interesting ones was the bioterrorism. And the other activities was he was the president of the Polish Society of Virology, and the research interest was the phytochemistry and virus culturing, the antiviral activity, and also the molecular biology technique in the assessment of the antiviral activity. Okay, without the future ado, let us warmly welcome Associate Professor Lucas Viantek. So, Professor, time's yours.
Okay, I hope my presentation is visible now. First of all, let me once again thank the organizers for inviting me here today. It is a great opportunity for me to tell you a little bit about my work. But first, before we even start, let me tell you that for me, it's very comfortable to be among you because I'm also a pharmacist. I've graduated pharmacy in 2006. But then I've moved to Faculty of Medicine. I've also worked in the field of medical laboratory biology. But still, there's something still in me, inside of me, from a pharmacist. So that is why I'm also interested in natural products. I did my PhD, and then following this, I did my habilitation in medical science, but still I am a pharmacist in my soul. So that is why for me, natural products are a very important topic because as the Honorable Dean already mentioned, we do have most of our drugs nowadays because of the study of natural products. We've known about it for many years. We've known about the alkaloids. We've known about Also anti-cancer drugs, which have been derived from natural bioresources. And we've also known that antimicrobial compounds can originate from natural products, as we have most of the antibiotics being natural products. So it was natural to also look for the source of antiviral compounds also among the natural products. And actually, when we look at the natural evolution of antiviral drug discovery, we'll also see some origins from natural products as well in this beautiful timeline, starting from the first antivirals, which were relatively toxic. They were thiosemicarbazones, for example, through the discovery of acyclovir, we will discuss in a moment. Also, the discovery of antiretroviral drugs that have some also origins in the natural products as well. Throughout the most recent discoveries of anti-hepatitis drugs, which are used in the treatment of, for example, hepatitis C, which also originate from natural products. A milestone in antiviral treatment was the discovery of iodexuridine. In 1964, ophthalmologist Dr. Herbert Kaufmann showed that this drug is actually useful in the treatment of viral diseases. It was the treatment of herpes virus-induced keratitis. However, this drug was extremely toxic. It was an anti-cancer drug. That is why it could not be used systemically to treat viral infections. It was only used topically. So was the another representative like 3-fluorothymidine. This was also used externally. Those two toxic, too many side effects to be given systemically. Also, the first smallpox drug, which is methisazone, it had limited activity and it was also very toxic. Then we had a breakthrough with the influenza treatment, the introduction of amantadine and imantadine, which were the first anti-influenza drugs. They were effective and they were relatively safe. However, nowadays we know that they've unfortunately lost their activity very rapidly because of the emergence of drug resistance. And then a huge thing in 1950s, Bergman and his colleague isolated a very unusual types of arabinosinol glucosides from marine sponges from Tessitaia crypta. And these drugs, these substances, this first important marine derived natural products were studied for anti-cancer activity, and they've shown a very promising biological activity, and they were also tested for antiviral activity. And once again, thanks to the discovery of these two natural products, spongothymidine and spongouridine, we had the development of vidarabine and cytarabine. And actually, these were the first marine-related compounds that were used in clinical practice. We had the antiviral drugs ARA, and then we had the cytarabine, which was used to as an anti-cancer treatment to treat, for example, different kinds of leukemia or lymphoma. These were not safe drugs. They had multiple side effects. This is true. However, this gave some chances of successful treatment of both viral and diseases and cancer as well. It also gave an opportunity to further modify them to obtain a series of semi-synthetic structural analogs. And finally, these allowed for the development of acyclovir, so acycloguanosine, which was the first safe antiviral drug that was commercially available, and we use it until today. In many countries, acyclovir is now available even over the counter. For example, in Poland, the acyclovir in the lowest dose of 200 milligrams is available as an OTC. Higher dosages still require prescriptions, but for this one, they are available for the patients without a prescription. Thanks to these studies, we also had the azidothymidine, AZT, which was further used at the beginning of HIV epidemics as an antiviral drug as well. Today we know that significant number of antivirals originate from nature, or we have them because of the study of natural products. This is the data, most recent data from 2020, which focuses on the period between 1981 and 2019, gathering all the possible data about the antiviral drugs and their origins. And still we see that there are some totally synthetic drugs, like 29% of them, but we also have the synthetic drugs, but which are based on pharmacophores derived from natural products, and this is 48%, and 6% of them are actually natural products. Among them, we also have biological drugs, which are getting more and more popular also nowadays. But still, the dominance is of natural products among these drugs. And this is an still ongoing process of discovery of these drugs, and still we see that significant number each year were natural products or substances which were derived from natural products. A great discovery also, and the huge advancement in the treatment of hepatitis, both B and C, was the discovery of these three drugs. First, let's start with hepatitis B. Nowadays, WHO indicates that there are two possible options for hepatitis B, for chronic hepatitis B. And of course, chronic hepatitis B is herein defined as having the hepatitis B surface antigen positive for at least 6 months. And WHO indicates the use of either tenofovir or entecavir for the chronic treatment of hepatitis B, and both of them are derived from natural products. Tenofovir is derived from natural product, and entecavir has a pharmacophore derived from a natural product. Of course, total eradication of hepatitis B among these people is highly unlikely, but still, the use of these drugs allows them for a complete suppression of hepatitis B virus replication and keeps them healthy and keeps them not as much contagious for other people because the level of the virus are dramatically decreased thanks to this treatment. And also, these people are not at risk of developing liver cirrhosis or primary hepatocellular carcinoma in the future. So we do have the possibility of management, maybe not cure, but management of hepatitis B thanks to these drugs. We also have the sofosbuvir, which was the first representative of the second generation of direct-acting antivirals used in hepatitis C. These drugs were introduced around 2014, so not so long ago, and they've revolutionized the treatment of hepatitis C. Sofosbuvir itself was not able to do it, but in combination with other drugs that became available in subsequent years, we have the success rate of over 95% in hepatitis C treatment. And this, in opposite to hepatitis B, is a complete eradication of the virus. So depending on the regimen, depending on the drug combination, and the state of the liver, the treatment might take 8, 12, or 24 weeks, but this is a complete eradication of the virus. And once again, this is possible also thanks to the study of natural products. Not to mention that tenofovir is also an antiviral, antiretroviral drug, which is used in the treatment of HIV, and still it is a part of a drug regimen for HIV-infected individuals. Another interesting story, a story of shikimic acid, which was isolated from Japanese star anise, in 1885. So this was a substance known to us for a very long period of time. However, it was also a base material used by Gilead Sciences in 1995 to synthesize oseltamivir. So also the current treatment for influenza virus is derived from a natural product. These are inhibitors of neuraminidase, enzyme which is necessary for orthomyxoviruses like influenza virus A and B for replicating, for being released from the infected human cells. So we have these new drugs, maybe not new because they have been around for some time already, but they are still used in the treatment of influenza all over the world and are derived from natural products. Another interesting story of betulinic acid. Betulinic acid also was known for us for a very long time. It was tested for multiple biological activities, and among them, it was also tested for antiviral activity and showed a remarkable inhibition of HIV replication. It was found that it is actually an inhibitor of the maturation process, which interferes with the GAG polypeptide of HIV virus. So this actually means that it stops the production of structural proteins of HIV virus. This is a unique activity. There are no drugs which have the same mode of activity. We have inhibitors of HIV proteases, but they work in a different way. So here was a unique mode of action. However, and this was a huge problem, it very rapidly induced drug resistance due to mutation in the nucleic acid of the virus. It very rapidly led to the loss of activity. That is why betulinic acid itself could not be used in practice. Even in clinical trials, it was shown that the activity is lost very rapidly. That is also why there were attempts to create derivatives of betulinic acid. We actually had two generations of them. The first one being bevirimat. It was developed by Panacos, and then it was sold to Myriad Genetics. And then, unfortunately, it was suspended. The further development of this drug was suspended because Myriad Genetics at this time was focusing on some other areas of biomedical research, and they did not want to invest further money in this project. So until now, it is on hold. But then we had the second generation, which was developed by Bristol Myers Squibb. It gave significantly better activity because of better tolerance to GAG polymorphism. So even some minor mutations did not influence the effectiveness of the drug. It was acquired by GlaxoSmithKline in 2015 during already undergoing phase 2b clinical studies, but unfortunately, because of gastrointestinal intolerability and also emergence of drug resistance, the further study was suspended. So here we did not have a significant success, but still it was a very interesting research, and maybe in future we will have some more even better derivatives of these drugs. Another remarkable story, a story of calanolidae. This is an example of sometimes great discoveries are made by an accident. There is a National Cancer Institute in the United States which finances scientists to travel all over the world and gather different kinds of natural products. And one of this was to Malaysia, to the island of Borneo, where they have collected a series of samples, and one of these samples, called Burley and Lee 351, was taken to the United States. It was tested for anti-cancer activity, but there was no activity. However, at NCI, not many people know about this, but there is also a subunit there that studies possible inhibitors of reverse transcriptase of HIV virus. The director of this unit was Stuart Legris until recently, and these samples, the same samples, were tested for the inhibition of HIV virus, and they have found a remarkable activity, a remarkable inhibition in the screening assay. So they've actually wanted to study it more closely. So they went back to Borneo. They went back to the same location they have gathered this particular sample, and they have found the area completely destroyed. I think it was about a developer building houses there or something like this, but they've destroyed the whole forest. And they could not find a single plant of this species on this area and on the whole Borneo as well. So it was an endemic plant, and they've actually completely destroyed it. Fortunately, a few existing still species were found in the Singapore Botanic Gardens, and this allowed for collecting enough material to isolate the compound responsible, and it was calanolidae, and for further biological studies. And they found that this is a non-nucleoside reverse transcriptase inhibitor, NNRTI, but having a completely different mode of action because it binds at two distinct sites in reverse transcriptase enzyme. With the first side, it is competitive, with the second, a non-competitive way. So it has a double diverse effect on the viral enzyme, and it was effective in vitro against also the strains which were resistant to other reverse transcriptase inhibitors like AZT. So it was a extremely promising discovery. It was also tested in clinical trials. Unfortunately, they found that the clinical effect was delayed, and also there were some severe side effects which made them discontinue the studies. However, it was a great example of a new type of biological activity. That is why many scientists try to synthesize derivatives with the same activity but limited toxicity. And such publications were published in 2008 and 2010, and then in 2012 with different kinds of derivatives, comparing them to antiretroviral drugs which are currently being used in the treatment of HIV. We also have a nice docking analysis that was done. And one of the study is originating from a very special team, which was from Rega Institute in Leuven. And this research group is led by Professor Jan Balzarini. This is a very famous person in the world of virology because he did a lot of research in reverse transcriptase inhibitors, and a great number of them is actually because of his work. And also, the team of Professor Jan Balzarini obtained a new scaffold from combination of the pharmacophore from calanolid A and also non-nucleoside reverse transcriptase inhibitor, which was already available and tested before in clinical studies. And they've came up with a completely new scaffold and then tested a whole series of different kinds of derivatives of this drug, and this was published in 2015. I had the great honor of listening to Professor Ian Balzarini lecture in 2014 and the conference in Sardinia, and he was mentioning then the studies they were doing with this type of compounds, and it was extremely interesting. A very, very nice and famous scientist. Naturally, the coronaviruses gained a lot of attention in the world of virology, especially after the first SARS-CoV-1 pandemics. And there were a lot of research done back then about coronaviruses, about anti-coronavirus activity as well. Many different natural products were tested. And at some time I was preparing a lecture actually about anti-coronavirus activity of natural products. And I was looking through literature and I found a very interesting compound and a very interesting activity of tylophorin from Tylophora indica, which was found back then. This was tested actually in 2010 as being a protease inhibitor of the coronavirus with a remarkably low IC50 value. This was interesting, and unfortunately, back then this was not taken further, but it allowed for the identification of possible drugs which were subsequently tested as an anti-coronavirus drugs during 2019 pandemic. Here we also have some studies of derivatives of tylophorin. Some of them had even better activity than the original tylophorin against the coronaviruses. And then we had a remarkable development here because tylophorin, this is from 2017, was found to inhibit also hepatitis C virus replication. We also had the combination of tylophorin with Janus kinase inhibitors with possible even better activity. And finally, also anti-tumor and anti-angiogenetic activity of tylophorin and promotion of apoptosis in cancer cells. This is quite interesting because from the historical point of view, we started the development of antiviral drugs from anti-cancer drugs, and here we have the opposite situation. We started with viral activity, and we ended up many years later with the anti-cancer activity. So this is also very interesting, but this additionally underlines how close these studies are together, the antiviral activity and anti-cancer activity. And this is because essentially we are targeting very similar properties. Both anti-cancer drugs and antiviral drugs, they inhibit often enzymes which are necessary for the replication of genetic material, either replication of DNA or production of messenger RNA or production of proteins as well. So we do have certain similarities here. And this is also why antiviral drugs are often extremely toxic. Acyclovir is relatively safe, but other drugs like ganciclovir or cidofovir are very toxic because they are also inhibiting the replication of nucleic acids in healthy cells, which are not infected with a virus. When you study antiviral activity, you must remember that these are obliged intracellular parasites. So viruses, by replicating inside the human cells, inside the living organism, are completely independent on the outside environment. They're incapable of replicating outside the human cells. And the only moment we can actually interfere with their replication is once the virus is already inside the infected cells. That is why it's difficult to achieve sufficient selectivity. And often the antiviral drugs, they do have significant side effects, as I've mentioned previously. Not every cell, of course, can be infected with a particular virus. The ones that can be infected and support the replication cycle of the virus are called permissive cells. But inside the human body, there are also non-permissive cells, which can be theoretically infected by the virus, but they do not support the whole replication cycle of the virus. And they usually do not produce infectious viral progeny, and we call such infections abortive. The viruses may influence the cells in a variety of different ways. Some viruses, of course, will be causing cytocidal infections, and these are most of the viruses we study in our laboratory. And thanks to the cytocidal nature of their infection, we can observe certain morphologic changes of the cell induced by the viral infection. They influence cellular physiology, biochemistry. They can also induce genotoxic or other biological effects. We can observe it, and this helps us a lot in the study of antiviral potential of different substances. However, the viruses may also induce chronic infections. And when they do so, they're usually much difficult to eradicate from the human body and to reach with the use of antiviral drugs, not to mention the latent infection, which are completely out of control using antiviral agents. Unfortunately, when the virus causes latent infection, there is actually nothing we can do because the virus does not replicate. And the antiviral drugs are only effective against the viruses which are replicating, not the ones which are latent. That is why we cannot eradicate a herpes virus once it successfully infects the human organism. Of course, additionally, we have some viruses which are causing transformation of the cells. We have some viruses which are oncogenic, like the Epstein-Barr virus or papilloma viruses. So they contribute to transformation of the cells, to cellular dysplasia, and potentially in the future to cancer transformation as well. There are many different ways you can culture viruses. However, the one most commonly used nowadays is the use of cell lines to support viral replication. So we have a variety of models supporting replication of the viruses. Some laboratories additionally use laboratory animals, but this requires appropriate conditions for these animals and also for the support of infected animals as well, which is another problem. And of course, this allows the permission of bioethical committees. So you need some results, preliminary results obtained in vitro to be allowed to test anything in vivo models. We also have the culturing, the possibility of culturing the viruses using embryonated hens' eggs, the oldest technique of culturing viruses, which is still used for some purposes like the production of vaccines, but not for routine culturing of the virus for scientific purposes like for antiviral studies. That is why we focus on virus culturing using cell lines. Of course, in order to do this, you must select appropriate virus and appropriate model for the replication of this virus. So we need permissive cells that will support the replication of the virus. We need to culture the virus to obtain high infectious titer to be further used in the research. We also need to study and then we may attempt to study their antiviral potential. First of all, we need to answer what type of activity we want to focus on, whether we want to test the antiviral or virucidal potential. In our laboratory, we are focusing only on antiviral activity, so the inhibition of viral replication in the infected cells. The virucidal activity is the interfering with the viral particles outside the human body, and such substances are used as disinfectants. So we do not study this type of activity in our lab. There are many different assays you can use for antiviral research. There are many different procedures you can be focusing on. In our research, most commonly we focus on cytopathic effect inhibition, so CPE assay. We also do yield reduction assay, most commonly using CCID50 reduction based on endpoint dilution assay. And we're also using molecular techniques like qPCR or RT-qPCR. We can also use neutralization assay or chemagglutination or detection of antibodies or antigens using ELISA or luminescence assay. This is, of course, possible, but we do not do this in the routine research on everyday basis. What we most commonly do is that we run our assays on the cell lines which are already infected with a virus. So we infect the cell line with appropriate virus. We do the pre-incubation for 1 hour. And then after removing of viral particles, after washing the cells, we add our potential antiviral substances. We study both natural products as well as synthetic compounds, but because of the topic of today's study, of today's lecture, sorry, I will focus only on the ones which are of natural product origin. Beside the influence of cytopathic effect, we're also measuring the infectivity, the reduction of infectivity of the virus. The most commonly used techniques for this purpose are either plaque forming reduction assay. So we measure how in percentage the reduction of the formation of plaques by the virus in the virus infected cells treated with the in comparison to virus control. We do not do this assay. We have a little bit different approach. We measure the reduction of the infectious titer of the virus using an endpoint dilution assay. So we actually collect the specimens from CPE assay. After freezing these samples to release the viral particles, we performed a series of dilutions. These are 10-fold dilutions, and then infect with each dilution the cell line. And then we observe how many cells in how many wells are affected by the virus infectivity. They show the appearance of cytopathic effect and compare it with the virus control. Typically, for this type of assay, scientists are using mathematical formulas. There are available calculators based on Reed and Minch technique, which allow to calculate the CCID50 value or TCID50. However, we do it in a little bit different way. We apply the measurement of cellular viability of the and then calculate the CCID50 based on the obtained results. And it showed a very nice effectiveness. And actually, the results we obtain are very similar to the ones obtained using mathematical calculations. That is why we just use them in the routine assays. So our protocol for study is as following. First of all, we study the cytotoxicity because we need to know what dosages will be safe for the cells to be used. We use different kinds of cell lines used for virus propagation. And subsequently, we also test the influence of these compounds on cancer cells, because we know that there is a similarity between antiviral and anti-cancer activity. So just in case, to cover also this part of the assay, we are also running them on the samples we are testing. Afterwards, we are doing the antiviral screening. The antiviral screening is based on the CPE assay. So we test how our compounds or extracts influence the virus-infected cells, whether we see some inhibition of the cytopathic effect. After 3 days, these plates are frozen and thawed usually 3 times. And after this, we do the potency evaluation. So we want to test how significant was the effect of the substance on the viral replication. And at this point, we are doing either endpoint titration or molecular analysis, or sometimes we do them both. It depends on how promising are the results are the screening results we obtain. We always start with cytotoxicity, just like in this example. For the cytotoxicity, most commonly we apply the most basic MTT test, which is based on the ability of viable cells to reduce the tetrazolium salt to formazan crystals. The higher is the biological activity, metabolic activity of the cells, the more formazan product we obtain, so also the more intensive violet color. We all usually have a very nice dependence between the concentration and the cellular viability, as in this example. And as you can see here, we've tested not only using a normal Vero cells, but also 3 different cancer cells, from hypopharyngeal, from colon cancer, as well as from adenocervical carcinoma. In this research, we've not tested for antiviral activity, but the screening for cytotoxicity is absolutely the same. We use the viral cell line for culturing most of the viruses we study. So based on dose dependence curves like here, we select the concentration which is not toxic for the cells, and then we use this concentration in subsequent antiviral assays. In case of the types of the viruses we study, most of them are cultured using the viral cell line, so the green monkey kidney cells. And for example, we have herpes virus type 1, human herpes virus type 1, also known as herpes simplex. Then we have Coxsackie virus B3, CVB3. This is an enterovirus, as well as adenovirus type 5. They're all replicating in viral cells. And in these 3 viruses, we have 2 of them which are DNA viruses. Herpes virus and adeno are DNA virus, as well as an RNA virus, because Coxsackie virus B3 is an RNA virus. As well as we have both enveloped and non-enveloped viruses, because herpes viruses are enveloped and adenoviruses is a naked virus without an envelope. We also study respiratory viruses. So we also have human rhinovirus here and human coronavirus 229E. The rhinovirus is cultured using H1 HeLa cells, and the coronavirus 229E is cultured using MRC5, which is a normal lung cells. Of course, for these studies, we have our own master cell bank. We store in our laboratory to provide us with a sufficient amount of cells for further research. Most commonly studied virus for us is the herpes simplex, the human herpes virus type 1. This virus is cultured in the viral cells. It is one of the viruses with highest prevalence in the human population. Most of the adult population is already infected with the herpes simplex, as well as this is a latent virus, so it establishes latency in sensory ganglia and undergoes reactivations throughout our life. Primary infection is usually in a form of herpetic gingivostomatitis. It happens usually at a very early stage of our life, and then we may suffer from possible reactivations. This virus causes labial herpes as a reactivation, but may also infect your eyes, leading to keratoconjunctivitis, which is extremely dangerous because it may even lead to blindness. It may also cause herpetic whitlow, which is also nicely presented here. And this virus, when it is introduced in the viral cells, we here have an example of a nice monolayer of viral cells, and after infection, we see the cytopathic effect. Usually it appears between 48 to 72 hours post-infection. Here we have Coxsackie virus B3, also normal cells and cells after infection with the cytopathic effect. And here we have lung cells, MRC5 cells, infected with the human coronavirus 229E and also the associated cytopathic effects. And this is how we can use it in practice. From my friend in Turkey, I've obtained extracts from Geranium peranaicum. We've tested it for antiviral activity, and we've found that the methanolic extract, especially the methanolic extract, suppressed the formation of cytopathic effect with a relatively low concentration range of 150 to 250 micrograms per milliliter. The replication was significantly inhibited. And then at lower concentrations of 100 or 50, we had the loss of antiviral activity. So this is an example of how a screening assay might look like. This is another research because we have obtained a promising results from a Geranium species from Turkey. I've encouraged my students in Poland to look for similar activity in Polish species. This is Geranium robertianum. And together with my students, I've did some research. And we've also found that some of the tested extracts, especially the one from obtained using hexane and ethyl acetate, showed a promising activity. We've also done subsequent fractionation of these cells. We've obtained 6 fractions from ethyl acetate extracts and also 4 fractions from hexane extract. And some of them also showed in the screening assay promising antiviral activity. We can also do real-time monitoring of infected cells. Using time-lapse imaging technique, we can acquire a series of photos after different periods of incubation and observe how the cytopathic effect develops in the infected cells treated with particular substance throughout the incubation time. And this is also an example of such an assay. In order to measure the impact of the substance on the viral replication, we do the titration assay. This is an example of research where we did not observe the reduction of viral infectivity, which is nicely shown here on the figure. The curves obtained for the virus control are almost identical to the ones obtained for extract-treated samples. These were all samples from Dandelion. These were obtained using different extraction technique. We've compared the Soxhlet extraction with the supercritical fluid extraction herein, but there was no antiviral activity. In opposite here, we have an example of titration assay for geranium samples we've mentioned a while ago. And in this case, we had a significant reduction of viral infectivity. For example, the methanolic extract at 250 micrograms per milliliter did not allow us even to measure the infectious titer of the virus because the cytopathic effect was present only at the lowest of the first dilution we actually tested. This is -1, and here there was some loss of cellular viability, but it did not continue. And here we have in delta log, we have the reduction of the viral titer, and for these 2 dosages, 250 and 200, it was above 4 log, suggesting us the significant antiviral activity of these samples. We can also use the molecular techniques. We can do it in the most basic way, so qualitative PCR with subsequent gel electrophoresis to show us the amount of the nucleic acid amplified in different samples. But this has very limited sensitivity, which is shown right here. These were samples I've tested like 13 years ago, so it was a long time ago. We are not using this technique anymore. But what is interesting here is that in this samples, which is a dilution of virus control, -5, -6, at -5, we still see some very, very light band, but when I actually tested these samples using real-time PCR, I found that there are still thousands of copies of virus present, and in the higher dilution, there was no band observed here, and still there were hundreds of virus copies detectable using real-time PCR. That is why we no longer use this approach. What we do are either semi or quantitative, qPCR. And this is an example of the study we did on Stachys sylvatica. We've tested those extracts against two viruses, the coronavirus and also the herpes virus. The coronavirus being RNA and the herpes being a DNA virus. That is why for the coronavirus, we have used the RT-qPCR, and for the herpes virus, we've used the qPCR without, of course, the reverse transcription. We did see some inhibition of 1.5 log for the coronavirus and 1.1 for herpes virus, but these are typically not relevant inhibition, because for a relevant antiviral activity, it is required for the virus to be reduced by at least 3 log. So this is an interesting activity, but not enough for sufficient suppression of the viral replication. But still, a very interesting result. This plant was from Kazakhstan. This is another study. This samples originated from Africa. Also samples from Africa, but the extracts were obtained in Turkey. So also it is a result of our cooperation with scientists from Turkey. This was from Spatoda campanulata. And for these samples, not only we have observed inhibition of viral infectious titer, but also we did the molecular studies. However, during the molecular studies, we have observed a very strange results, I would say. The cytopathic effect was inhibited. Then when we did the titration assay, we also observed very high inhibition, above 5 log, although at very high concentration of 500 micrograms per milliliter, but it was a sufficient inhibition of the virus infectivity, and even at half lower concentration of 250, still there were over 4 log inhibition. But when we did the molecular studies, and this was actually a typical quantitative analysis, because we have used a in vitro certified diagnostic test for herpes virus to prepare a calibration curve we have subsequently used for amplification of viral DNA. We've observed very little inhibition of less than 1.5 log. So I was a little bit at a loss of how to interpret the data. And I thought that maybe the biological effect we observed, so the inhibition of viral replication, is a result of not inhibiting the viral DNA replication, but inhibition maybe of some late stages of virus replication, like the production of the proteins or the assembly of the infectious particles. That is why I've contacted my friends in Turkey and asked them to perform a docking analysis for me. I've selected a couple of substances from this extract and 3 of the key enzymes of herpes viruses, and asked them to do the docking analysis. And what they found is that, in fact, most of the tested substances were inhibitors of viral proteases, which explained it all, because proteases are necessary for the late stages of viral replication, so the production of structural proteins, for example. That is why we observed the inhibition of the infectivity, because there were no infectious particles being produced. but the viral genetic material did replicate in the cells, but was not allowed further to progress into the release of viral progeny. So this is also an example how we can combine different kinds of biological assays to achieve the goal, as is to look for the antiviral properties of natural products. So we found that actually the substances from Spatoda campanulata are the inhibitors of viral proteases. We've also tried to check whether the Spatoda campanulata samples managed to inhibit the replication of other viruses, and we did it against the Coxsackie virus B3, and there was no activity. So the antiviral properties of these plants were very specific, only against the herpes virus. The things to consider when you are planning this type of research, of course, involve the proper plant identification. I'm not a botanist. I'm always relying on other people to accurately identify the plant and also to store the appropriate vulture specimens in their resources for other people to confirm the species or to confirm the results we obtain. We also always do the chemical analysis. I have friends who are running the LCMS or doing GCMS to identify the major compounds and the metabolites which are found in the tested extract. Sometimes we are also doing isolation of compounds or fractionation of the extracts, as was shown in the results I've presented a while ago. Things to consider, important things are the solubility, and this is a problem often, what to use and how to dissolve the samples which are derived to us. Most commonly, we use dimethyl sulfoxide, but not every extract will be able to dissolve in DMSO. And also some people do not want to use DMSO because it may produce some artifacts when the samples are stored for the prolonged period of time. Also, the solvents used for the stock production may influence the cellular viability or influence the biological effects we study. So this is also another things to consider. Another thing is testing volatile compounds. A number of times we were asked to test the essential oils or extracts containing volatile substances. And this is always a problem because we incubate our cells in 37 degrees Celsius, which means that a lot of these compounds will be evaporating over time, and we are losing them during the incubation period, and it is completely unavoidable process. And additionally, the influence on cell cycle. When we study viruses, they replicate within the cells. So sometimes when the replication of the virus is inhibited, we must ask ourselves whether this is a result of the inhibition of certain cellular processes which do not allow the virus to replicate, or the suppression of the viral replication. It's difficult to answer this question without doing detailed studies. Concerning the volatile nature of essential oil, this is another very interesting study. I really enjoyed it. We've done some time ago with volatiles. I was always interested in what is actually happening with volatiles during the experiment. So I've asked my colleague to give me some essential oils and also volatile compounds, and we've incubated them in the cell media with or without cells. And then after some periods of time, collected samples, I've isolated them. I've isolated the volatiles using hexane, and then my friend did the GCMS analysis to check the amount of major volatiles. And as you can see, we have an enormous decrease in the amount of volatiles. Here we have the essential oil from eucalyptus, and you can see how rapidly these volatiles are disappearing from the tested substances. Another interesting observation is the blank at the end. on each plate with experiment, we always have a blank. So wells without the cells, supplemented only with cell media. And I've collected samples from these blanks, and actually we have identified volatiles in this blank, which means that the components of the essential oil not only evaporate from the plate, but also evaporate and dissolve in the wells which are around the tested wells. This may hugely influence the results which are obtained when essential oils are being tested. So this is also a very nice observation. We also attempted to produce microemulsions and emulsions of the essential oils. This was a complete failure, because the emulsions we have obtained were more toxic than the essential oils, which were actually tested before. But one thing which was interesting about this research is that when you're dissolving something in a biological solvent, in DMSO or any other solvent, you must take into account the toxicity of the solvent itself. Here, when we tested citronella, eucalyptus, or mint oil, we have found that these microemulsions always had a higher toxicity than the essential oil dissolved in DMSO. And when we have taken all the microemulsions and showed them in the single figure like this, they were identical, which means that actually here the solvent was toxic and not the essential oil, which is pretty interesting. I know that I'm running late, but I just want to show you 2 more slides and it will be over, I promise. We've also did the influence on the cell cycle, and this is an experiment I've done using a image cytometer like this one, which allows for the cell cycle analysis. So this is what happens with cell control after 5, 8, 13, etc., up to 72 hours of incubation. We can observe it on these histograms very nicely when the cells divide and when they complete their replication, when they produce the monolayer after 72 hours. And these are some of the samples I've tested for antiviral activity. And actually, I found that in their case, there was a significant increase in the number of cells which were in sub-G1 phase, which were probably apoptotic. So this shows us how important it is to also include this type of study in biological assays to confirm the influence of substances on the cell cycle or apoptosis or any other cellular process that may interfere with the observed antiviral effect. So I hope that within this short presentation, I've shown you not only the significance of antiviral research and the role of natural products in antiviral research, but I've also shared my enthusiasm of studying natural products and a little bit of research from my own laboratory. And finally, I would like to thank my co-workers, especially Professor Barbara Reiter, who taught me all that I know about cell culturing and about working with viruses, etc. Unfortunately, Professor retired this year, and she's no longer helping us, and we are left alone. But hopefully, we will try to keep up the good work and also continue the antiviral research that was going and still is going in our laboratory. So once again, thank you very much for your attention.
Yes, thank you very much, Professor Sviantek, for such a stimulating and insightful lecture. Yes, the depth and the clarity of which you explored the antiviral products and prospect at the natural products have certainly inspired us among our participants. Okay, now we will move on to the Q&A session. So for the audience that like to ask the question live, so you can raise your hand, and I will invite you to unmute your microphone, or you can just feel free to type your question at the chat box, and I will read it to Professor Sviantek. Okay, we have two big question here, sir. First from Zainur. Yeah, he about to ask you about how do you see the integration of the ethnopharmacological data with the uh, combination and pharmacophore-based virtual screening to accelerate the discovery of the antiviral products.
Ethnopharmacology is a very interesting topic, and I've actually been reviewing for the Journal of Ethnopharmacology for a while. I was invited by Professor Viloyen to review some papers over 10 years ago for the very first time, because there were a lot of papers sent to the Journal of Ethnopharmacology, which were testing the antiviral properties of indigenous plant species based on the observations or interviews or data collected from natural healers about the application of various plants for example, skin diseases that may result from viral infection. So this is a fact. This is an ongoing process. However, interestingly, most of these studies originate from traditional Chinese medicine. There are a lot of samples sent, manuscripts sent testing the combinations of traditional Chinese medicine drugs in the form of granules, etc. However, I always am a little bit skeptic about this work, because very commonly they're testing incredibly complex matrix of secondary metabolites. So within a single plant species, we may have dozens of different secondary metabolites, and they are testing products containing dozens of different plants. So it means that the amount of secondary metabolites within this matrix, within this sample, is incredible. It's very difficult to predict which one, why they are active. There's so many possible combinations, interactions, or synergisms to take into account that it's virtually impossible. So I am a little bit skeptic about so complex matrix being tested, but this is a fact that ethnopharmacology provides us very important data on the use of natural products by the traditional medicine, by these healers. And testing them is important, but we have to progress further when we observe the significant biological activity of these products. We need to test also where this actually comes from. So at some point, it will be necessary to isolate the compounds, because this is the only way these will be available as drugs. Only when we isolate the compound, when we describe exactly how they work, not as a matrix built of thousands of different compounds, but a selected compound or a group of compounds at least. So this is my opinion about this. You've also mentioned screening and drug banks or plant banks, if I remember correctly. And this is also important, but I am a skeptic about this as well, because this was especially seen during the early stages of COVID-19 pandemic, where we had an overflow of papers on inhibition of viral enzymes. So many people either did the docking analysis or simple inhibition assays towards enzymes of plant extracts or compounds. And this actually is interesting. However, this has to be subsequently repeated using in vitro assays because it provides very limited information. The fact that potentially something inhibits an enzyme in silico studies does not mean that we'll observe any activity in vitro studies. So this is another problem for me.
Yeah, thank you, sir. Okay, for the next question was the as we know that the natural products that often possesses highly flexible structure. And how does this structure flexibility influence the design of the pharmacophore model when the screening for antiviral targets, such as viral proteases or polymerases, kind of things?
Flexibility, you mean
Yeah, the flexibility
stability?
structure, flexibility chemical structure. There is, yeah, we know that some the metabolite, the secondary metabolite that have the variety of the secondary metabolite that we can, modification with the modification the structure to.
I do understand now. So when we find a possible hit in a screening assay, so potentially antiviral activity, of course we can do chemical modifications, we can obtain derivatives and then test their activity using biological assay. This is completely normal and this is done all the time. I think that is what you were asking, or did not,
Yeah.
or maybe I did not understand it correctly.
Okay, so we can proceed for another question. So it seems like which the in silico or the in vitro technique that the best support for this experiment about the viral receptor selection and the drug design.
Well, I think the best model to start the experiments is using the cell lines, as I've mentioned. First, use the screening assay to check the influence on the cytopathic effect. Then look for how strong was the actual effect by measuring the reduction of either viral titer or viral load. So either test how the extract decreases the infectivity of the virus, or test how many viral particles are being produced based on their nucleic acid. This is the best approach. However, the antiviral studies, they not only take a significant amount of time, but also they require a number of resources being spent. So typically, first we do the screening assay. If we observe a promising effect in the screening assay, we progress further. Sometimes we stop at the screening assay. If there is no influence on the cytopathic effect, we usually don't progress further. Unless these are like very precious or very promising samples because of some additional biological studies that were done on these samples. Then sometimes we do the infectivity. But in most cases, we only test further the samples which give us a promising biological activity.
Okay, sir. Thank you. For another question, sir. From your experience, what are the key success factors in collaborating about the natural products into the antiviral candidates for the preclinical evaluation?
Well, first of all, find a colleague you trust and who will stimulate you to do the research, and also who will contribute for this research to be published. Because I had so many examples also with the cooperation from the scientists from abroad that sent me samples, I've did the study, and then nothing happens then. So these results are either put in a drawer or presented on a conference or two, but there is no success in publishing the papers. This unfortunately happens. But if you find someone who is eager to not only send you the samples, but also take it further and publish the results, it doesn't matter whether these are great results or just moderate results. For me, it's important to publish them because every result, even the one which does not show the activity, is a progress because you show other people that it's not the way to go. They should look for some other plants, other extracts, other ways of testing, etc. So in order not to repeat the experiments that were unsuccessful. So for me, that is important. And also, the key fact is to be happy with the research you're doing. So also look for the people you like to talk to. It makes cooperation much easier.
Okay, sir. Yeah, and I have read several of your papers, and then I was amazed that you have a collaboration with so many scientists outside the Poland, and you can get the samples from Turkey, you can get the samples from Kazakhstan, and many others. And as yeah, amazing work. Okay, there's any participants that will ask for the Professor Fiantek directly? Maybe you can put in a chat box. We have approximately 5 minutes. Okay, yeah, another question, sir, about how that the relationship between the virus and the boosting of our immune system.
Yeah, I've seen the question, kill the virus
Mhm.
or boost the immune system. That is an interesting question. And well, it depends. When we talk about killing the virus, we talk about actually virucidal activity, so we can inactivate or otherwise kill the virus outside the human body. However, once the virus is inside us, it is not possible. We do have some substances which are interfering with the protein surfaces of the virus, and some of them are used for HIV, for example. But most of these drugs are actually effective once the virus is already inside the human cells. So they are antiviral but not virucidal. It is true that most of the drugs we have are actually inhibiting the viral replication in the human cells, so they're typically antiviral. But there were also some attempts to use the immune system against the viruses, especially in relation to the viruses which hide in the human body. And this is true for the HIV, which is not typically latent virus, but it does hide in the human body. And it's also true for all the herpes viruses. There was even an attempt to create so-called kick and kill theory. And this means that we stimulate the viral replication and then we try to eliminate this virus from the human body. But also boosting the human immunity is crucial here, especially the cellular immunity, which is necessary for the eradication of the virus. And that is also why whenever we have a cell-based immune deficiency, we see not only very dangerous course of primary infections with viruses. Now, this is what happens in HIV patients. That's why they suffer from recurrences of herpes. That's why they suffer from herpes zoster reactivations. That is why they suffer from cancers like non-Hodgkin's lymphoma, because during these cancers we have activation of Epstein-Barr virus. That's why they suffer from Kaposi sarcoma because of the reactivation of Kaposi sarcoma human herpes virus. But also the immune system in the latent infections caused by herpes viruses is crucial. We have infections with herpes viruses after graft transplantations. After kidney transplant, the reactivation of cytomegalovirus can lead to complete destruction of the transplanted kidney, for example. And in this way, boosting the cellular immunity or otherwise controlling the immunosuppression is a crucial way also in prevention of these complications among immunosuppressed individuals. We also have some antiviral drugs which are actually increasing the immunity. They're available also in Poland as OTCs. So this is also a way of treatment or activating the human immune system to help us fight with the viral infection.
Okay, sir, that's fascinating. Okay, for all, we have now reached the end of our public lecture. So we are now proceed with the symbolic certificate handover to our esteemed guest speaker, Professor Lucas Piantek. Yes, sir. Pardon us that we are give you the the digital certificate for this event. Thank you, sir.
That is perfect. Thank you very much once again for inviting me, for the possibility to meet you, to see you. Thank you very much once again to the dean and for the moderator for nice introduction and for helping us to give my speech and to have the wonderful contact with you. I would also like to mention that I am a little bit shocked by the number of people attending this lecture, over 200 people. This is really, really nice. And I would like to congratulate you about being able to gather so many people, so many participants. It was an honor to me to have this lecture today with you and to spend my afternoon with you. Thank you very much.
Yes, thank you very much, sir. So on the behalf of the organizing committee, I would like to express our deepest gratitude to Professor Lucas Viatek for his generous time, brilliant insight, and inspiring presentation. So we hope that today's lecture has broadened our understanding about how natural products that are basically rooted from the traditional medicine and the modern science hold promising potential as the antiviral therapies in both current and future of the public health challenges. So to all participants, thank you for your active involvement and curiosity. We hope that this session has sparked new ideas and the potential of the research collaboration. So we can conclude that, especially that, so you can kindly fill out the feedback form that shared in the chat, and a certificate for participation will be available for those who complete the form. And we'll also encourage you to stay connected to future lecture and research discussion from our faculty. So once again, thank you for your participation. Stay safe, stay healthy, and keep learning, everyone. So at the end, I will say Assalamualaikum warahmatullahi wabarakatuh. Good day and see you again soon. Thank you, Professor Spiare.
Bye-bye.